Hi, I'm Jingyi from University of Medicine. Thank you for your interest in our poster. This is a walkthrough of our work. Redox balance, the balance between production and elimination of oxidants, is disrupted in osteoarthritis. So monitoring cartilage redox balance has potential for disease staging and treatment screening. Given the close relationship between mitochondrial function and cellular redox balance, evaluating changes in mitochondrial membrane potential is a way to monitor cartilage redox balance. Measurement using fluorescent dye show that mitochondrial membrane potential is responsive to mechanical loading. However, the translational potential of exogenous dye is limited. Alternatively, optical redox imaging allows label-free real-time evaluation of redox balance. ORI uses the autofluorescence from NADPH and FAD, which are cofactors in cellular redox reactions. The fluorescent intensity corresponds to accumulation of the cofactors, and previous study from our lab showed that ORI is also responsive to mechanical loading. Despite the potential of ORI as a clinical tool, the underlying mechanism of altered ORI matrix has not been studied thoroughly, and the relationship between ORI and the established redox measurements such as mitochondrial depolarization remains unknown. So the objective of this study is to develop and test the predictive model of mitochondrial membrane potential from ORI following mechanical loading. Full-thickness patella cartilage was cut into thin strips. A pair of strips was glued to a macroscope-top loading device, and one strip underwent tensile loading at either 100% per second or 10% per second speed until 30% ultimate strain. The other one was unloaded control. Optical redox imaging was performed before and 30 minutes after loading. For each fluorescent channel, the intensity in each imaging location was normalized to the baseline intensity in the corresponding zone. After ORI, both strips were stained using JC1 dye and imaged in each cartilage zone. Polarized mitochondria was stained red and depolarized ones were green. Mitochondrial membrane potential was described by the red over green intensity ratio. ORI matrix at 10 and 30 minutes after loading were correlated against the mitochondrial depolarization level using Spearman's rank order correlation. A linear mixed effect model containing fixed effects, random effect, and all-way interactions was used to predict mitochondrial red over green ratio. Significant fixed effects and random effects were retained in a new model, which was trained using the training dataset, which is 70% of the full data, and evaluated using the test dataset, which is the remaining 30%. These two figures show the correlation between normalized autofluorescence intensity in each channel and the mitochondrial depolarization level, as described by red over green ratio. Mitochondrial depolarization level was significantly correlated with both green and damping intensity. For the linear mixed effect model, significant fixed effects were green intensity, damping intensity, loaded versus control group, and loading rate. The random effect of sample was also significant. The model that retained all significant predictors resulted in an average difference of 8% between the predicted and actual red over green ratio across 10 datasets, in which the full dataset was randomly split into training and evaluating dataset for each trial. In summary, the results show that ORI matrix can be used to predict mitochondrial membrane potential after mechanical loading. To move forward, findings in this study have to be expanded to measure to distinguish disease states to further advance ORI as a clinical tool. Thank you again for listening, and please reach out if there are any questions or comments.